primary importance it is. Proteins are the most abundant biomolecules of the living system. So when you observe the living organisms, so proteins are most abundant in that one. The main source of the proteins are nothing but milk, cheese, pulses, isn't it? Peanuts, fish and meat, right? But generally what happens, people think that meat will have what? More amount of proteins, but pulses will have more amount of proteins when you compare with meat actually. The problem arises between these two. What happened? It cannot be digestible. Meat is not digestible, but the pulses are what? Very good digestible. And milk also what happened? Having more amount of proteins, but at the age of what happened? Old age, otherwise what happened? Something around more than 20 years. Not only old age, more than 20 years, milk cannot be digestible. Milk can be digestible only by the small children's. So best source for the proteins will be what? Pulses only. Pulses and peanuts. These two are the main, isn't it? Sources for us to get the proteins. Next, they are present in every part of the body and from the fundamental basis of structure and function of life. So to sustain our life, to function our life, the part of the protein is what? Very important. And the growth, maintenance of the body, proteins are very, very important. Next, very important thing is all proteins are polymer of alpha amino acids. I told you in the last class, what are alpha amino acids, beta? Anybody? What are alpha amino acids? Anybody? Which amino acids are called alpha amino acids? Yesterday we discussed this one. What are alpha amino acids? Which can be called alpha amino acids?
Amine group is directly connected to alpha carbon.
Amine group is directly linked with what? A carbon which is bonded to what? Carboxylic acid, which is called what? Alpha carbon. Classification of the proteins. On the basis of the structure, based on the structure, we can classify the proteins here. Now let me see the two classifications. One is fibrous, other is what? Globular. Fibrous and the globular. Best example for the fibrous is what? Anybody can give one example for the fibrous? Is it possible for you? One example for the fibrous protein. Bio students know? Give one example for the fibrous and globular.
Keratin.
Keratin means what? Where it doesn't. In our general example, you say. Don't
Sir, in hairs.
hair. Fibrous generally what happen from straight like that. Globular will be look something what happen? Round shape like that. Any example? Globular?
Insulin.
Yes. Yes, beta. You are saying something?
Insulin.
Insulin, egg. Egg also will be one of the best example. Egg. Egg looks what happened? Round shape. Globular. Hair. Something like this. Now, why like this? We explain here. In detail, let me see. Now, here in this case, chain of amino acids lies side by side. They lie side by side like this. Okay. Now, chains of amino acids fold. They are folding like this. To give globular structure like this. To give a globular structure like this. Hair, isn't it? Looks like something like this only. Egg looks something what happened like this. Egg means what happened? That part. Next, structure of peptide bonds in protein already we discussed. Again, I am repeating, peptide bond or peptide linkage is an amide formed between C double bond O NH2 group by elimination of water molecule. Already we discussed this one. Now, this is the first amino acid I have taken. This second amino acid we have taken, already we have taken, and have we removed the water here, and we got actually a peptide bond like this. Now, you are calling this one as what? A peptide bond here, C double bond O NH2, isn't it? So, when one or more molecules of amino acids combine with dipeptide, it forms tripeptide. Now, if it reacts with another second molecule, now it is a peptide bond. Take another one, we'll get dipeptide like this. Already we completed this. Next, let me see the primary structure here. The first type, primary structure. The primary structure of proteins refers to the sequence of amino acids held together by peptide bonds. So, how do we get the primary structure, beta? Anybody? How do we get the primary structure? Because of what? Peptide bonds. The primary structure because of the peptide bond. One important thing is that you cannot change the primary structure of any protein. Any protein, if you observe here, whether it is keratin or anything, albumin, we cannot change the primary structure. Now, what is primary structure? For example, I am writing something, glycine. Next, what happened? Alanine. Next, what happened? Valine, isn't it? Again, alanine, glycine, valine. This is what actually these polypeptides are made upon by glycine, alanine, valine, glycine, valine, like that. By what happened? By removing water like that, like this, isn't it? Something like this. Now you cannot change what happened, the basic structure, the primary structure. So primary structure is formed by peptide bonds. The secondary structure of the proteins, if you observe, long amino acid chains exist in the loop or coiled or stretched or folded. Something because of something coiled type of structure. How it will get? Because of hydrogen bonding. Let me see that. A particular shape of protein molecule which refers to a secondary structure. So whenever amino acids, once we got a structure like this, glycine, alanine, isn't leucine something, they undergo something stretching, folding, coiling. So that makes what actually? Secondary structure. So two different types of structures will be there in secondary. One is alpha helix, other is called beta pleated sheet section. Now, chain gets coiled by twisting into right hand spiral known as alpha helix structure. Let me see this one. We'll see. Alpha helix. This is what, like the spiral binding. You see what happens? Spiral binding, you have seen the same like that. So here what happened? It coiled like that. Alpha helix we can call this. This is called what? Alpha helix. Now when you observe here what happened, what is this hydrogen bonding? Where we get this hydrogen bond? Already I told you what is peptide bond? C double bond O NH. Correct, beta? This is called peptide bond. Now it can show hydrogen bonding with another peptide bond, isn't it? It can show hydrogen bonding with another peptide bond. So because of the hydrogen bondings, this type of coiling, folding is taking place. So in any peptide bond, because of hydrogen and oxygen, they undergo hydrogen bonding. So they are folding because of what actually? Hydrogen bonding. This is what actually these chain get fully stretched or extended, resulting in a flat sheet like that. When you stretch this one, what happened? You will get a flat sheet like this. The best example is a phone wire. Now this is a sheet-like structure. Something. It will get a sheet-like structure. You have seen now just now because of this flat stretching that one will get like that. When these chains are placed above, one above the other, above and up below, they give what now? Beta sheet. Now you got beta now. See, when you take more number of chains like this here, more when these chains are placed one above one, isn't it? They give what? Beta pleated sheet section. Examination, nobody asked to draw what happened, beta sheeted. Nobody asked you. For your understanding only we have taken. Next, what happened? Tertiary structure of the proteins. Now, the tertiary structure represents folding of polypeptide chains further the further undergo folding here. Now you what happens, something unfolded, something you see what happens. Now folded like this, because of what happens, something folding like this. You got this one. The polypeptide chains are held together by what happened? For example, if you see what happens, sulfur, can you give one example for sulfur containing amino acid? Give one.
Methionine and cysteine.
Very good.
Methionine and cysteine.
Methionine and cysteines are the best example for sulfur containing what actually? Amino acid. So disulfide linkages, isn't it, will make you what happen, further folding like this. So what type of forces are operating now here? So van der Waals forces generally operates for any molecule, even if you take what happened, methane, hydrogen, nitrogen, all molecules will have van der Waals forces. So to know what is van der Waals forces, if you don't know the van der Waals forces, you just watch my video in states of matter, force of attractions. I have already made a video, isn't it? You watch that one, you will get what is van der Waals forces. And hydrogen bonding, everyone knows about hydrogen bonding. Disulfide linkage because of having sulfurs in the amino acids. All these forces, isn't it, which are reason for what happened, for getting what? Foldings actually. Okay? All these forces of attractions are reason for the what? Because of the folding. Further folding taking place. Now, next, finally, what happened? Quaternary structure of the proteins. The proteins composed two or more polypeptide chains are referred to as subunits. Quaternary structures refers to the spatial arrangement of these subunits with respect to each other. This is what spatial arrangement. The final arrangement, it is what spatial arrangement of the subunits like this. Now, this is what actually you are having alpha, you are having beta. Alpha sheeted, beta plated structures. Here, 4 subunits of proteins are arranged in a single unit like that. All are what? Arranged this like you got an arrangement you got. For example, I will show you what is how you get tetrahedral shape when hybridization taking place. So you have 4 hybrid orbitals are there. You have 4 hybrid orbitals are there. And these 4 hybrid orbitals will arrange in the form of tetrahedral like this. So now we got a tetrahedral shape. So in this way, what happened? The final, isn't it, spatial arrangement, you got something what happened as a quaternary structure. Right? Primary structure unfolding is giving secondary, further folding is giving tertiary, again further folding is giving quaternary. So 4 structures are you having: primary, secondary, tertiary, quaternary structures. Take summary of this one. Primary, the sequence I told you, no, glutamic acid G, glutamic acid GLU, okay, arginine, phenylalanine, glycine. This is a basic structure you cannot change. This is because of what? Polypeptide linkages. Because of polypeptide. If I take glutamic acid, arginine, phenylalanine, glycine, if I mix these, what happens? I get a basic structure that you are calling as a primary. Secondary, that is because of what? Alpha helix and beta sheets like this. Alpha helix, this one is a, when you arrange these one adjacent like this, parallel like this, we get beta sheets. Now this one is a 3-dimensional structure we are going to get in the tertiary like this. This one will be arranged like this. And lastly, what happens? Spatial arrangement here. Spatial arrangement after complete what happened, everything, hydrogen bondings, everything what happened, we get what happened, this type of things. Right? This is about the primary structures. Can anybody give example which of the following contains nitrogen beta?
Proteins.
Proteins only, isn't it? Right. Secondary structure of protein is coiled, linear or zigzag structure beta. Secondary structure.
Coiled.
Very good.
Coiled.
This is very important beta. And generally what happened, what type of questions you may get in proteins here? Nobody asks you what happened, draw secondary, beta plated, nobody asks you, leave it that one. What type of questions do you get in that one? Albumin, something what happened, what we call globular and fibrous. From that only we will get questions. Examples, globular and fibrous examples will get. Now let me see this one. US of the protein. They are found, already we know this one, leave it this one. They are found in the living cells, in the skin, hair, everyone knows this one. Okay. And nerves, everything we have. As hormones, protein regulates the metabolic process here. To regulate the hormones, what happened? We need the proteins here. Isn't it? For regulating the hormones, proteins are compulsory. Proteins form major part of the immune system. Then, for example, if you are suffering from any disease, otherwise any fever, what happened? You need to take what happens, some proteins for that one. To get what happened? To strengthen your immune system. Isn't it? Nowadays, what happened? Corona, what happened? Patients, what happened? They're taking what happened? Some protein food. Why? Because to improve the immune system. Actually. Protein helps in general wear tear of body organisms, isn't it? For making that body organisms, what happened? Building block of that organism, what type? Proteins are very, very important. And they maintain what happened? Body temperature and metabolism also what happened because of what? Proteins. And the growth because of the proteins. These are what? General use of the proteins. Actually. Very important beta. What is denaturation of the proteins? It's a very, very, very important point for us. How do you make denaturation of the protein? Then that means changing the quaternary tertiary structures, isn't it? You are changing the structures of the protein, not primary structure beta. Protein is found in a biological system with a unique three-dimensional structure and biological activity is called a native protein, general protein, a natural protein it is. Now, when a protein, when it's a natural system, you are changing by what? Changing the temperature and pH. So whenever the temperature of the protein, pH of the protein changed, then what happened? The protein hydrogen bonds will be disturbed. So that whatever that structures, the tertiary, quaternary, secondary structures will what happen? Changes. In that case, what happened? You are calling the denaturation of the protein. This is what definition is. By how you are changing the beta? How you are changing the structure of protein? What is how you can change the structure of the protein? What are the possibilities? Tell me. By changing the pH and also what? Temperature. This is very important beta. You need to remember this one. Due to this, globulus unfold and helix gets uncoiled. So coil becomes uncoiled, and that is the reason what a protein lost its biological activity. So whatever the function of the protein will lose here. Let me take some examples for this one. During the denaturation, secondary tertiary structures are destroyed, but not the primary structure. This is useful for what? Incorrect or correct statements like that. Primary structure you can't destroy by using what happened, changing the temperature. Now, when you observe the egg, the coagulation of egg, isn't it? Egg white, especially egg white, on boiling is common example for the denaturation, isn't it? When you boil the egg, what happens? It will coagulate, isn't it? Simple, whatever, precipitate. So this is nothing but called denaturation. So you are eating, how you are eating the egg? By denaturating the proteins, isn't it? So it is one of the example for what? Denaturation. Another example, what happened? Making curd from the milk. Making curd from the milk by adding what happened? Little bit of lactobacillus, something what happened? Lactic acid or lactobacillus, something we can call. So by adding what happened? Lactic acid. That means what? But what actually? From what? Curd. Isn't it? From the curd we just add that. So already I think you know coagulation of egg is what type of process beta? Denaturation of the protein actually. This is now this is about what? Proteins. I will again explain in the textbook also. Just now finally we'll take what happened. Now remember enzymes are not there in the syllabus. You don't read enzymes for CBSE. And what happened? That last topic, vitamins also not there in the syllabus. Enzymes, vitamins are not there in the syllabus. Now let me take what happened? Nucleic acids. What are nucleic acids? These compounds are the substances of heredity. Generally what happened? It will come along with what? Heredity. From ancestors to what? From ancestor to us, how we get. From parents to how we get actually. Nucleic acids resembles proteins even though chemically they are different. Just like they are what proteins, but they are chemically what completely different. because they contain sugars. Sugar they contain actually, nucleic acids. The polynucleotide chain, polyester chain is the backbone of the nucleic acid of molecule. So this is the backbone. Let me see this one, the structure also. The slides. Ester of phosphoric acid with sugar. We'll see the structure. So it contains what actually? Ester of polyphosphoric acid with sugar. Now what are the sugar and polyphosphoric acid? Let me see in the next slide. The nucleus of the cell, the chromosomes are made up by nucleoproteins, which contains two types of nucleic acid, RNA and DNA. Ribonucleic acid, deoxynucleic acid. Ribose nucleic acid, deoxyribose nucleic acid. Now let me see the structure of this one. This is a ribose beta. The general just how you draw the fructose structure, cyclic form. How you draw the structure like glucose form, isn't it? Just like ribose is also we can do. This is what actually furanose structure, because it's a 5-member ring. So furanose structure here, just like fructose here. I've taken what beta D ribose because OH is here. On which direction beta? Upward direction. Therefore it is called what? Beta D ribose. D ribose because second carbon, the OH is on right hand side. The second carbon OH is on right hand side. That is the reason I have taken like this. Now this one, if I take what happened, how we can form the nucleic acids by using this one. This one is what? Beta deoxyribose. This one is beta deoxyribose. You observe here what happened. What is the difference between these two you are observing beta? Ribose is a carbohydrate beta. What is ribose here? Carbohydrate, monosaccharide. Simply say monosaccharide. Example what? Monosaccharide I have taken. Correct? Now, what is the difference you are observing beta between these two? This is ribose, this is deoxyribose. That means deoxy means what happened? Here we don't have any oxygen. You lost one oxygen. In DNA, the oxygen is removed from the second carbon atom. You observe here what happened? Here oxygen is removed in where? DNA. We don't have the oxygen over here actually. So one is ribose, other is what? Deoxyribose. See, in DNA, deoxyribose, in RNA, ribose. Here we are not getting oxygen. Structure of nucleic acid. A chain is made up from many nucleotides like this. You'll see that. Every nucleic acid is made up by what? Sugar. That is nothing but carbohydrate. Nothing but what? Ribose. Nitrogenous base phosphoric linkage. Now we see what is this phosphoric linkage here. Let me see this phosphoric linkage now. Now, nucleic acids are two types beta, pyrimidines and purines. So these you have to remember in exam. I think you know this already. In biology they will come: cytosine, thymine, uracil, guanine, adenine. Adenine structure, don't remember adenine structure beta. Nobody asked in examination, write adenine structure here. This is what guanine structure. This is cytosine structure, thymine structure, uracil structures. And these are the structures of what? Bases which are present in the nucleic acids. DNA is a chain made up by nucleotides joined together by phosphate ester linkage. Let me see this ester linkage. You see here. A base, just now we seen here. Just now we seen all different type of bases here. Adenine, guanine, like that here. Sugar means what? Ribose. Now this is what? Phosphate linkage here. This is the phosphate. Every, isn't it? Nucleic acids, if you observe what happened, they look something. Base, sugar, a phosphate, ester linkage. This is called ester linkage, beta. Phosphate ester linkage. In this way, what happened? You have basic structures like this. Now, how they are joined with each? A, adenine, isn't tyrosine. All these are actually we have taken the bases here. Structure of the DNA and RNA. They are joined like this. Now, why the DNA appears helix structure? Because of coiling like this. That is because of what? Hydrogen bonding here. Double helix structure. DNA and RNA. The structure of DNA is double helix in which two strands run anti-parallel like this, something like this. One is something like this, resulting into right-handed double helix due to hydrogen bonding. The helix, the coil, because of what? Hydrogen bonding. This one, you know, this one DNA means deoxy. This is no matter. This one. Anybody? What is this? Uracil?
Pyrimidine.
Right, very good. What discuss on textbook better, because in this slides what happened, you can only make structures here to appear. It will appear easy for you, because textbook, if I what happened, open what happened, it could not appear what happened easy. Now, with this what happened, biomolecules are completed. And I told you already, surface chemistry, I will make videos. Okay. I will put in a playlist called Surface Chemistry. You can watch that video whenever, if you have free time, you can watch that. Now, one thing you have to remember, beta, amino acids in which what happened, remember this star mark. Star mark indicates what, beta? They are actually what?
Essential.
Essential. So you just remember only essential only, and remaining will be what will automatically become non-essential here. So try to remember this one, isn't it? Valine, leucine, isoleucine, arginine, lysine. Next, what happened? Threonine, methionine, phenylalanine. It came in 11th class, I think, no, in bio. These are came in 11th class, I think, in bio.
Yes, sir.
And also similarly acidic and basic also. Remember that one also. Zwitterion definition is important for you. Definition of zwitterion, you have to remember. Now, what is meaning of peptide bond? Very important beta. What is the definition of peptide linkage, peptide bond? You have to show like this in examination. Okay. Now, penta, tetra already you know, then more number of amino acids are combining, you will get like that. Now, you read this beta, fibrous proteins. Okay. So they are formed because of when the polypeptide chain run parallel and held together by hydrogen bonding, disulfide bonds. Other what happen? Van der Waals force or something. You will get what happened? Fiber-like structure here. So main examples there. One more thing, beta. Do hair is soluble or insoluble, beta? In water. In water. So remember what happened? Insoluble. This is very important, beta. Fibrous proteins are insoluble in water. Very important thing it is. Fibrous proteins are insoluble in water. You see keratin. So where it present keratin? Hair, wool, silk. They never soluble in water. Even you heat also, they don't soluble in water. Myosin present in the muscles. So these are the examples. All of you please remember these examples. They may come definitely in examinations here. Keratin and myosin present in hair, wool, silk. This one is in the muscles here. This is important for you. Insoluble and examples are important for you. Next, globular, because of looking globe like that round shape, they call like that here. They are soluble. They are soluble. Example, you take an insulin, albumin present in the egg, isn't it? That comes under what actually? Globular proteins. So you have to remember what? Whether soluble or, because incorrect statement they are asking this one. Globular proteins are insoluble in water. They give like that. Incorrect statement it is. Next, primary structure, secondary structure. इसमें कुछ ज़्यादा नहीं रहता. आपको alpha helix एक बार पढ़ो. Beta plated structures यहाँ. This is what primary, secondary, tertiary, quaternary structures that is there in the textbook. Now, next, denaturation of the proteins. Denaturation of the proteins, you have to remember the definition first. Definition first. And how we change occur? By changing the temperature and pH. Physical changes. Best example we have taken coagulation of egg and converting milk into what? These are what best methods. By just changing the pH and temperature. How egg? Coagulation can be done by what actually? Temperature only. Correct or not? Enzymes are not there, beta. Leave it enzymes here. Don't read enzymes here. Okay. Vitamins also not there. Leave it the vitamins actually. Actually, what happened, wherever easy topics are there. In chemistry, easy topics are actually vitamins and everything. They removed chemistry in everyday life. Very, very easy topic, beta. Most of the students will pass by reading what happened. Chemistry in everyday life. That chapter was deleted. Polymers was a very simple thing. Even a 6th class students also can make answer for that. So that was also deleted. So why? Because I'm stressing the easy chapters in which you can score marks. They are deleted now. There are no more. Even maths isolation of elements is very simple. All these are theoretical chapters were deleted, which are very simple for average students. That is the reason what happened. You try hard now. Today onwards, what happened? Read whatever topics we are discussing. Read that one. This is what nucleic acids you have taken. Ribose and deoxyribose I have taken, isn't it? Now these are the different type of what happened. Bases which are present in the DNA. What are the bases present in the DNA? They may ask in examinations better. DNA contains 4 bases: A, G, C, T. You remember like A, G, C, T. A, G, C, T in DNA, right? And the fourth, the first 3 bases are same as in DNA, but the fourth one is what? Uracil. This one. Now nucleic acid structures we can easily remember: nucleoside, nucleotide, in which we are having what? This ester, phosphoric ester linkage is there in the nucleotide. Where in this case we don't have this phosphor ester linkage is not having in this one. Now after the DNA RNA, the DNA fingerprint, generally useful in the forensic laboratories, isn't it? And this is in hormones are not there, but leave it this way. So this is about the biomolecules. So there will be number of questions will be there here. All these are what questions actually. So try to answer these questions here. Whichever not enzymes, you leave it this one. Topics of enzymes, disaccharides, polysaccharides, importance of the carbohydrates. All these are vitamins. They are not there. Remaining you can practice. With this what happened? We have completed, isn't it?